The accurate prediction of, all atom biomolecular structure is really the holy grail of modern biology. It's absolutely central to understanding life. And maybe more importantly, it guides rational drug discovery. For years, that quest was almost exclusively focused on proteins.
Exactly, just proteins.
Yeah.
But in the last few years, these deep learning models, especially AlphaFold3, AF3, have just blown the doors open. They've expanded their reach to cover this whole spectrum of entities. We're talking nucleic acids, small drug-like molecules we call ligands, even ions. This ability to predict across different domains is, well, it's revolutionary.
But when you revolutionize a field that fast, you create a new kind of problem, right? A kind of chaos. How do you actually know which model is the best?
Precisely. We just didn't have a fair, systematic way to test these new capabilities across all those domains at the same time. I mean, how do you rigorously compare AF3 against its main competitors, Boltz-1, Chi-1, HelixFold3, Proteinix, when the targets range from a tiny molecule to a massive piece of RNA?
And that is our mission for this deep dive. We're going to unpack the answer, which comes from a new paper that introduces something called FoldBench. This is not just another small study. This is the big, exhaustive, all-atom benchmark the community really needed to find out who's generalizing and who's just remembering.
And FoldBench is robust by design. It's made of 1,522 real-world biological assemblies, and they're sorted into 9 distinct prediction tasks. It was built specifically to stress test these models and, crucially, to map out where they fail.
So the immediate takeaway for you listening right now is this. AlphaFold3 is the consistent leader. It shows superior accuracy across most of these tasks. But, and this is the critical insight, the evaluation reveals these persistent major limitations, especially when the models have to deal with structures they have genuinely never seen before or structures that are incredibly flexible. That memorization effect is a huge vulnerability.
To really set the stage here with FoldBench, we have to understand the scale and, I guess, the rules of the game. Previous studies were always siloed. You get one paper on peptides, another on protein-ligand interactions, and you just couldn't compare the results. They used different metrics, different data sets. It was impossible.
And the biggest problem for any structure prediction benchmark is stopping the model from essentially cheating. If AF3 has already seen 99% of a protein structure during training, predicting it isn't really generalization, is it? It's just recall.
That's the core technical brilliance of FoldBench. They use what's called a low homology filter. They were really aggressive about filtering out any structures that had high similarity to the model's training sets. They focused on PDB entries that were deposited after January 2023, which was AF3's training cutoff. So if a model does well here, it's truly generalizing. It's not just regurgitating what it already knows.
And that strict filter makes the diversity of the molecules they tested even more impressive. This is a massive, varied set.
Oh, absolutely. It's broken down into monomers, so that's 334 proteins, 15 RNA, 14 DNA, and then you have the crucial interactions, the interfaces. We're talking 279 protein pairs, 172 antibody-antigen complexes, a huge set of 558 protein-ligand targets.
Yeah.
And then, various protein nucleic acid and protein peptide assemblies on top of that.
So you put all that diversity in front of the models, and AF3's overall lead across the main metrics, DocQ, RMSD, LDDT, that becomes the main story. But wait, before we keep throwing acronyms around, we should probably clarify what these scores actually mean for anyone listening who isn't a structural biologist.
That's a great point. It's vital. When we say a prediction has high accuracy, we're usually talking about a couple of things. First, there's RMSD, or root mean square deviation. You can think of this as the average distance error. It's the distance between the atoms in the prediction and the true atoms from the experiment. A low RMSD, like under 2 angstroms, means your prediction is very, very close to reality.
Okay, so RMSD is that global distance error. What about LDDT?
Right, LDDT, local distance difference test, is a different flavor of accuracy. Instead of just that global position, it looks at the local environment of each atom. An LDDT score goes from 0 to 1. So if you score, say, 0.88 for a protein, that means almost 90% of the local atomic geometry is correct. It's a measure of local quality.
And the last one, DocQ, which you use for interactions.
Yes, DocQ is specifically for docking problems, like a protein connecting with another protein. It basically synthesizes those other metrics into a single score that tells you if the interface, the exact spot where they connect, was predicted correctly. A high DocQ means the binding is right.
Okay, so with those definitions in hand, we know AF3 is the leader. But is it a total sweep, or does the competition still have a few tricks up its sleeve?
It's definitely not a sweep. While AF3 is the most robust general predictor, The study found some very specific task-related strengths in the others. For example, HelixFold3 actually came out as the strongest performer, specifically on protein-peptide interfaces. It hit an 89.5% success rate. So that means if your specific question is about a peptide binding to a protein, AF3 might not actually be your best first choice. The right method really depends on the biological question you're asking.
Let's pivot to the segment with probably the most immediate commercial relevance: protein-ligand interactions. This is the bedrock of modern drug discovery, right? Finding that molecular key to fit the protein lock.
It is. And for the whole protein-ligand set, AF3 did get a solid 64.9% success rate. And remember, success means the ligand's position error, its LRMSD, was less than 2 angstroms and its local quality was high. That's a strong baseline.
But this is where the findings kind of shift from being great news to a major warning sign. The study found that even when the protein pocket itself was unseen, so low similarity to the training set, the protein modeling was still high quality. The protein wasn't the issue.
That's the critical discovery. The bottleneck wasn't modeling the protein's overall shape. It was correctly placing the small, flexible ligand inside it. And the data shows this crystal clear dependency. The docking accuracy for the ligand gets noticeably worse as the ligand's similarity to the training set goes down.
So the model has a great memory for binding pockets it's seen before, but a pretty poor imagination for how a brand new molecule might actually fit in that pocket.
Exactly. It's really powerful evidence that these models, and that includes AF3, are relying substantially on memorized binding modes they learned during training. They're great at generalizing protein folding, but they show really limited generalization when you give them a truly novel, unseen ligand.
And that limitation becomes a critical failure when we talk about more sophisticated drug design, specifically targeting what are called allosteric sites. For anyone unfamiliar, why are these sites so important for modern medicine?
Allosteric sites offer much greater therapeutic value because the ligand binds away from the main active site. It subtly changes the protein's function without just blocking the main entrance, which usually gives you much better drug selectivity, fewer side effects. But FoldBench showed the models fail here badly.
Can you give us a real-world example of how that training bias makes the prediction fail?
Sure. Look at the case study with ADP binding to a kinase, PDB8FNY. This molecule binds to a distinct allosteric pocket. Most of the predictions just completely failed. They either missed the pocket entirely or put the molecule in with the wrong shape. But even more telling was the result with a popular target called CDK2. All the models incorrectly placed the ligand directly into the main orthosteric active site. That's the primary site where the protein's natural partner, ATP, is supposed to bind.
So if you're a scientist trying to design a breakthrough drug for a new allosteric site, the AI has this built-in bias that says, nope, I know a better place for that, and it just defaults to the main active site it saw over and over in its training data. That feels like a massive flaw for innovation.
It is. The model defaults to what it knows best. And what it knows best is the most common binding site. It dramatically limits the discovery space.
Okay, let's move from small molecules to these huge complex interactions, protein interactions or PPIs. These things mediate almost every function in a cell. Here again, AF3 is still leading the low homology set with a 72.9% docking success rate.
And you see that same structural memory effect here too. The accuracy for PPIs goes up steadily as the structural similarity between the target and the training data increases. If the geometry of how the two proteins connect is novel, the prediction quality suffers.
But AF3 did show a little glimpse of being able to break out of that recall pattern, right? In a really difficult case, the domain-swapped homodimer 8-DPA.
That's right. This complex requires a huge conformational change to form. And AF3 successfully modeled that swap state. Not only that, it ranked the correct dimer as the best prediction among all its samples. The other models completely failed. They just got stuck on the monomeric or non-swapped state. So it shows AF3 has this capacity for modeling flexibility, which is very exciting, but it's just not reliable yet.
All right, now for the frontier that proved to be the toughest for every single model: antibody-antigen complexes. These are so critical for developing immunotherapies, but they're defined by just extreme structural flexibility.
And they are notoriously difficult because of that flexibility in the CDR loops, which are the binding sites. AF3's success rate just dropped significantly here, down to 47.9%.
Wow, so the best model failed more often than it succeeded.
Exactly. A failure rate over 50% across all current methods is just too high for reliable therapeutic design.
And what's the biggest structural reason for that? What's the bottleneck?
It's the CDR H3 loop. It's the most flexible and hypervariable region of the antibody.
Yeah.
The analysis showed this really strong negative correlation between the overall docking accuracy and the quality of how the H3 loop was modeled.
So a tiny error in predicting that floppy H3 loop can just cascade into a total failure to dock the antibody correctly.
Precisely. In one of the case studies, PDB-8PIH, a tiny error, the H3 loop RMSD, just moving from 0.17 angstroms to about 3 angstroms, caused a complete collapse. The dot Q score plummeted from a near-perfect 0.96 all the way down to a useless 0.03. A small local error just destroys the whole model.
The study did mention that increasing the number of guesses or samples did help AF3.
It did, which is good, but this also exposed another fundamental weakness that affects all the models: ranking. You can generate more and more diverse structures, but the model's own confidence scores often fail to reliably pick out the one correct near-native structure from the dozens of samples. If your ranking algorithm can't tell the good from the bad, then generating more samples is just a waste of time and energy for the other models.
And on the antibody front, they also broke it down by complexity. Nanobodies, these are smaller single-domain binders, they did a bit better, right?
Yes. AF3 got a 53.3% success rate on nanobodies versus 45.4% on standard antibodies. It's not a huge leap, but it does highlight the advantage of having less complexity. Nanobodies have fewer moving parts, fewer chains interacting. It's just a slightly easier puzzle for the model to solve.
So let's wrap up with the area that showed the most general underperformance across the board: nucleic acids. DNA and RNA structure prediction has really been struggling to keep pace with the advances in proteins.
The numbers are pretty alarming. When we look at that local quality score, the LDDT, the average for RNA and DNA monomers is somewhere between zero point 0.2 and zero point 0.6. You have to compare that to protein monomers, which are reliably hitting up to 0.88. Nucleic acids are just not being predicted with anywhere near the same reliability.
And why are they so much harder? Is it just their size?
It's a combination of size, flexibility, and complexity. Large RNAs, like this aptamer 7ZJ4, are huge, 374 nucleotides long. All the models failed to reconstruct the global architecture. You're getting massive RMSD values, around 20 angstroms. They just can't get the global fold right. And even locally, things like GC-rich nucleic acids that form complex G quadruplexes. They're also extremely difficult. Was there any good news at all for nucleic acids? Yes, actually, specifically when they interact with proteins. AF3 did much better on protein DNA complexes, a 79.2% success rate, than it did on protein RNA complexes, which was 62.3%. And that higher performance for DNA is probably because its structure is more predictable, that canonical double helix, and also because there's just richer training data for DNA-binding proteins. So if we synthesize all these gains and gaps, you get this picture of AF3 as a very robust leader, but one that is really vulnerable at the point of novelty. The benchmark clearly highlights this model memorization problem in data-scarce areas like ligands and nucleic acids, and this real difficulty with highly flexible systems like antibodies and those allosteric sites. The path forward is pretty clear from the paper. We urgently need to expand the scale and diversity of our training datasets, maybe using data synthesis. But even more critically, we have to develop better scoring or ranking algorithms. The current unreliable confidence scores are the biggest bottleneck. They completely neutralize the model's ability to leverage all the diverse structural samples that it can actually produce.
The dream of AI in structural biology has always been the ability to generate completely novel structures for new drugs, to show us something we haven't even thought of yet. But what these FoldBench results show is that our best models are still highly, highly sensitive to how much a target molecule looks like something they've already seen.
Exactly. The current state of the art excels at recalling the past.
So if true breakthroughs in drug design require finding molecules that bind to novel sites or adopt completely new shapes, and our best AIs can't even confidently tell us if their own predictions are correct, how do we move beyond just recalling the past? How much longer will structure prediction struggle to truly validate the unknown future of molecular architecture? That's really the question the community has to wrestle with now.